Gita Kolluru. Gita did her PhD and master's work at UC Riverside, so not too far from here. With, I think, David Resnick as her master's advisor, and then Marlene Zook is her PhD advisor. She, by the time she finished, had published several papers with them and other faculty at UCR. She then did a postdoc with me here for several years, and we also published lots of papers together and had a lot of fun. And from then she went on to do a lectureship at Humboldt State for a couple of years, and then got her current faculty position at Cal Poly, San Luis Obispo, where she's now a full professor. And I recently got to visit her lab, and it was great to get to meet all of her students. She has a really vibrant research program, all with master students and undergraduates at Cal Poly. So Gita's research, I think the primary focus of her research is sexual selection, so animal behavior more broadly, but more specifically sexual selection, so things like mate choice, dominance interactions, and how that affects the evolution of things like coloration. She is, I would say, pretty much a specialist on Poeciliid fishes. So most of her projects, going way back to her master's research, have been on Poeciliid fishes, several different species. But she's worked on other systems too. She has papers on sand crabs and one on rattlesnakes, and also, I shouldn't forget, crickets and their parasitoid flies. So she has some taxonomic breadth to her research, but again, Poeciliid fishes are her main focus. So let's welcome Gita and give her a chance to give her talk.
Thank you. Thank you all for coming today, and especially nice to see some familiar faces, and I look forward to meeting those of you I haven't met yet, and I really enjoyed interacting with people that I've chatted with already. A huge thanks to Greg for inviting me today. These are some action shots from in the field in Trinidad from when I did my postdoctoral work with him, as he mentioned, and Greg gave an excellent talk in our department when he came to visit, and I'm really glad that he got to interact with people in my lab, and I'm happy to be here as well, so, and meeting members of his lab, which is great. So, color polymorphism is among the most dazzling aspects of biological diversity. And here I'm talking about the persistence of discrete genetic color variants in the same population at a rate above the mutation rate. And this phenomenon requires an evolutionary explanation. Why doesn't the one best morph take over the population, or why doesn't speciation occur, right? So those are kind of the ends of the continuum. What maintains these multiple morphs in a natural population? And typically the answers to those questions come from some aspect of balancing selection. This list is by no means exhaustive, but these are some of the big contenders that people usually turn to. Some type of negative frequency dependent selection, such that rare morphs enjoy high fitness until they become more common, at which time their fitness declines, and that can maintain polymorphism. Or some type of spatially or temporally or spatiotemporally fluctuating selection, such that different morphs experience different fitness advantages depending on what kind of microhabitat they're in, or depending on temporal aspects of that population. And then, because color is involved, oftentimes explanations involve some sort of opposition between sexual selection and survival selection acting on those color patterns. Melanin polymorphisms occur in a number of taxa and are especially intriguing to me because melanin polymorphisms are implicated in a number of other correlated traits. And so, as Ducrest et al. Showed in this awesome paper back in 2008, the melanocortins that are in part responsible for deposition of melanin in skin and in feathers bind to the MC1R receptor, which is typically what mediates the deposition in skin and feathers. But there are also other melanocortins. This is a pleiotropic gene, the POMC gene, that leads to variation in correlated traits, including things like mating behavior, aggression, and immune system function. So it's likely that in melanin-based color polymorphisms, there are going to be covariates that help to maintain that polymorphism in natural populations. So our lab works with, as Greg said, Poeciliid fishes, and as probably many of you know, Poeciliids, at least New World Poeciliids, are characterized by being internal fertilizers that give birth to live young instead of spawning. So male Poeciliids have a copulatory organ that's called the gonopodium. It's a modified anal fin, and when males mate with a female, the male has to rotate that gonopodium forward and insert it with really good aim into the female's gonopore, her genital opening. And this all happens very quickly, as I'll show you in a slide coming up. And that's one big characteristic of these fish. This happens to be the species I worked with for my master's. I try to include it whenever I can, just for fun. And then these fish give birth to live young, so they don't spawn. Approximately every 20 to 30 days, depending on the species, females produce babies, live young. These are a bunch of newborn fry. And females can store sperm in these species typically. So if a female mates with a male, even just one male, she can store sperm and produce offspring for months sometimes, depending on what kind of sperm storage has happened. So a number of Poeciliid fishes exhibit melanin-based polymorphisms. Here are representatives from 3 genera, just to show you the kinds of polymorphisms I'm talking about. Typically they involve barring or spotting patterns, and oftentimes the melanic males in these species are rare, and I'll talk more about that coming up. Our lab works with one of these species so far. We're focusing on one species, and that is Gyrodinus metallicus, which always sounds like a heavy metal band to me. It has a common name, the metallic live bearer, but I don't know of anyone who outside of the fish hobbyist world that uses that name. So Gyrodinus is a Cuban endemic genus. There are 7 species. They're all endemic to Cuba. And although excellent work has been done by some Cuban researchers on this taxonomic group, not a whole lot has been done outside of those Cuban researchers. And so not a lot is known about this genus, really, including this species. So most of the males of Gyrodinus metallicus look like this. This is what we call the plain morph male, and they look really similar to the females in terms of coloration. They're dimorphic for that gonopodium, but they don't have any coloration that's different from that of females. And this is by far the most common morph in the wild, but there are rare melanic males that we call black morph males, and you can see that they have this ventral black coloration that extends to the gonopodium as well. We don't yet have morph frequency data from the wild, but what we do have are excellent collaborators in Cuba, including this person who I'll name in a later slide, Rodet Rodriguez. He and his colleagues have surveyed a multitude of sites in Cuba where these fish occur, and report to us that the melanic males are found throughout the island in all of the populations where the species occurs, and that they're rare. Maybe something like 10% or fewer of the males are these black morph males. I'm happy to tell you that when I've taken fish from the wild and looked at their behavior and their morphology in general, it's really similar to the populations that we have in the lab that I'm going to talk about today. So that was really important to see. Of course, I look forward to being able to do field work in Cuba and quantify some of these traits there, but for now I'm happy that these fish exhibit the same kinds of patterns that we see in a lab setting. So here are here's the distribution map for Gyrodinus metallicus, and it occurs throughout Cuba, as you can see. And this species tends to occur in disturbed lowland areas. So these are not pristine mountain streams. These are kind of ditches and swimming holes and things like that. They vary to a certain extent in how disturbed they are, but primarily disturbed areas, all with high predation. Got to work in a couple of those sites. So this is the Facunda Lake in the Zapata Swamp area in the southern part. Both of these are in the southern part of the main island in Cuba. And both of these sites had melanic males, the black morph males, but the vast majority of males were plain morph males. And then this is a swimming hole. It looks really beautiful in this image, but it's a very disturbed site. Lots of people swimming there and drinking and partying in the same little area as we were trying to do work there. So very disturbed areas with lots of, the bottom of these sites has a lot of heterogeneity from the natural environment, but also from stuff that people have tossed in these areas, right? So this is not G. metallicus, it's another Poeciliid, but I want to highlight that some really good work has been done with this species. This is the eastern mosquito fish, Gambusia holbrooki. And in this species, there are also rare melanic males that have this striking melanin pattern, but the vast majority of males, again, are the silver morph. And Joe Travis and Lisa Horth and their colleagues have done some excellent work in a number of different axes looking at this species, and have shown that the melanic males are selected against by females, so females hop away from them, basically swim away from them when they're trying to mate. And when the melanic, and they're also more aggressive, by the way, so which is not surprising given the pleiotropic connection between aggression and melanin that I talked about. So when these aggressive melanic males become more common in the environment in a population, their survival declines, suggesting that there is a negative frequency dependent selection mechanism keeping these melanics rare when they occur. They are thought to suffer from a lower predation risk than the silver males, and that may explain why they don't go extinct in natural populations. So today I'm going to talk about work that our lab has done most recently on G. metallicus, and I'll address these 5 questions, and I'll take each of these in turn. Before I do that, I want to emphasize that when I say we, I really do mean they. So these are my main collaborators, my former and current graduate students, Erin Woyen, Crystal Castillo, and Samantha West. All of the work I'm going to talk about today, or almost all of the work I'm going to talk about today, is master's thesis work that they did. And then some of that work is in collaboration with Dr. Sue Bertram at Carleton University, and this is Rodet Rodriguez in Cuba. And so he is an amazing biologist who really knows almost everything about the Cuban freshwater ecosystem, and was an excellent guide down there. He is now doing his PhD here in the States, and so it's wonderful for him and unfortunate for me that he's not in Cuba, as much anymore. But it's great that we have this collaboration ongoing. And then I really want to emphasize that all of the data I'll present today are data that are collected largely by undergraduate students. So Cal Poly is a primarily teaching institution. We have no PhD students. Rarely there are postdocs, but not in my lab and not typically. And so the work is overseen by master's students, and then really undergraduate students drive our research. So part of my goal today is, of course, to talk to you about the research that we do, but also if anyone's interested in what this kind of job is like, please feel free to chat with me about it, and I'm happy to talk to you about it. Our students are amazing, and by far the best part of my job. And helping to mentor them and guide them through the research process is what kind of keeps me going to work every day. Absolutely. So it's the part that I enjoy the most. And this kind of work, I don't want to minimize it by saying that it's undergraduate student driven, because these students produce publications and publishable data, so which is fantastic. They also present at conferences, and it's a very good system that can be rewarding in multiple ways, including based on the metrics that we usually use, getting grants and publishing and going to conferences and presenting. So all of those things happen with undergraduate students driving the system. I also want to talk about where we got our fish from, because one of the constraints we operate under is that we're working with a Cuban endemic species. It's really difficult to export fish from Cuba. It's difficult enough to do field work, to even go to Cuba, and to do field work there, but to export them is tough. So a lot of the fish that we had, at least in the past, were fish that were from a stock that I got from David Resnick's lab. That was unusual compared to the wild, because it was entirely black morph males. Right? This is really different from what occurs in the wild. And so we have that stock that we have been working with. That stock has been captive for years, long in captivity. So keep that in mind as I talk about results, but it's amazing that he is a sort of a repository for all these Poeciliids that are tough to get, tough to acquire and maintain, and he wants to ensure that people continue to do research with them, which is great. So that's how we acquired all these fish. But we're fortunate that more recently, Rodet and his PhD advisor, Dr. Ingo Schlipp, were able to get some fish exported from Cuba to send to us. So we also have a more recent Cuban stock that we just got a couple of years ago. So I'll be also talking about data that are from descendants of those fish. And this was a mixed morph stock, so it had the black morph males and plain morph males, and so much more reflective of what there is in Cuba. So the first thing we want to know is, do these morphs differ in behavior? That's the fundamental question when we're talking about this kind of a polymorphism. And so to address this, I'll first talk to you about the ways that Poeciliid fishes transfer sperm, because this becomes one of the most important axes of behavioral variation. So in general, Poeciliid fishes can transfer sperm in one of two ways. One is typically called a sneak copulation. So this is a plain morph G. metallicus male, and you can see it takes less than 1/10 of a second for this whole thing to happen, for him to, and it's called a sneak copulation for a reason, right? The male watches the female from afar. It's been described as lurking. He lurks in the vegetation, watches from afar, and then when he sees a female, he oftentimes chases her in what appears to be kind of a very violent kind of chasing behavior, really different from the other strategy I'll show you. So he chases the female around, and then oftentimes without her cooperation copulates with her. And so he inserts the gonapodium without doing any kind of a display beforehand and transfers sperm that way. And of course, there's post-copulatory sexual selection probably operating in this species. No one has yet looked at that, so it is not guaranteed that this kind of a mating would produce offspring, but it happens quite a lot, and especially in plain morphs, as I'll show you. The other alternative for Poeciliid fishes in general, and for those of you who know about guppies and things like species like that, the other alternative is for the males to do a display. So here is a male G. metallicus doing a display. The courtship display is what I'm going to call it for now. This display of males is different from something like a guppy. Guppy males swim around near a female, and then they do a very discreet display, and then they stop displaying. The G. metallicus display, so the male expands his dorsal fin, he holds the gonapodium away from his body, tilts his chin up, and seemingly is showing off his color patterns to the female. It's not a discrete point in time that he displays and then stops. He does this continuously. Males will do this for minutes, maybe even hours at a time. I haven't pushed it to see. Pretty much it's all males do. I don't know how they survive sometimes, because they don't seem to stop to eat or do anything else, right? Females spend the majority of their time trying to get away from males. Okay, that's pretty much the system. And some Poeciliid females, like guppies, give a behavioral signal when they like a male's display. Unfortunately for us, in G. metallicus, there is no such behavior on the part of females to show that they like the display. However, what we do have is that the females are extremely aggressive to males whose display they don't like. So the fact that a female doesn't aggressively bite and chase the male away is kind of the best indicator we have that she likes his display, potentially. Right? So being able to make copulation attempts, as you have seen in this video, he's made a couple of copulation attempts, and one of those may have been successful even. The fact that the female is permitting him to do that is a good measure of the fact that she's potentially interested in him as a mating partner. So one of my grad students, Crystal Castillo, undertook a study in which she put size-matched males of each morph together with a female in direct interaction tests. This is part of a larger study that I'll talk about more coming up, but I want to talk about the main difference in behavior between the two morphs first. So she performed a series of such observations to see whether there's a fundamental difference in terms of their courtship behavior or their mating behavior. And here's what she found. This is in fact a color-associated behavioral polymorphism, because the display is performed almost exclusively by black morph males. In these 20 pairs, only 2 of the plain morph males ever performed what looked like a display, and it was for a pretty short duration. So this is almost exclusively the display is performed by the black morph males. Plain morph males sneak copulations almost exclusively. All right, so very big difference in their mating behavior. So we then wanted to know whether sexual selection favors one morph over the other. There is, for all intents and purposes, what appears to be a courtship display, and we've called it a courtship display in the published literature, and others have as well. So we wanted to know, is there evidence that sexual selection favors that black morph over the plain morph if those males are displaying to females and giving them the opportunity to exert mate choice, whereas the plain morph males aren't? So we performed standard dichotomous choice tests. These involve a tank setup where there's a male on either side, an area compartment for the female, a neutral zone, and then we look at the amount of time the female spends associating with each of the 2 males. We size match the males in every case so that we minimize the size difference between them, because we want to see whether females have a morph preference without a confounding effect of size differences or a size preference, right? And we also randomize which males on the left first and which males on the right first. And we always perform 2 trials where we perform this trial and then switch the males and test the female again. And the order in which she sees the black morph on the left, et cetera, is randomized. And in this case, these females had been individually isolated for several weeks prior to being tested in this association time test. I just want to pause here to say I used to be skeptical of association time tests because I thought, oh, what if a female just kind of strays into one of these zones and we're calling it mate choice? Typically what we see is the two fish are interacting at that partition, seemingly trying to get to each other, right? So it's not subtle usually when a female likes a male on one of these sides. And if it's a transitional going into one zone versus the other that doesn't last a threshold amount of time, we don't use those data. So, and a female who seems disturbed and kind of swimming erratically around her compartment, we don't consider that mate choice either. So I'm now convinced that this is in fact a good measure of mate preferences. So here's what we found. Contrary to our prediction, females appear to prefer plain morph males based on this association time. They seem to prefer plain morph males over black morph males. This was surprising. However, when we parse this out into the two groups of females, remember I said that we had the stock fish that I got that had been in captivity for a longer period of time that are black morph biased. These are females who have seen only black morph males their whole lives, have only had the opportunity to mate with black morph males, versus females from the more recently acquired stock from Cuba who have seen both morphs during their lifetime. When we parse it out that way, we see that this preference is driven by these females in the black morph biased group. So it's possible that these females who were raised only with black morph males may prefer novel males that they hadn't seen ever before because they'd never seen plain morph males before in their entire lifetime. And in fact, in the recent evolutionary history of that stock of fish, there hadn't been plain morph males. So the females that are more indicative of what's happening in the wild seem to exert no preference, morph preference, based on these data. So Crystal and her team also then put all three of those fish together and performed those direct interaction tests I talked about earlier. In addition to looking at the amount of time males spent following females and displaying to females, they also looked at copulation attempts and then some measures of aggressive behavior. Some of these are obvious. Gonopodial display involves a male extending his gonopodium and showing it off to another male. So they use their gonopodia not only in the context of mating and displaying to females, but also in male-male competition. They show off their gonopodia to each other. They also use them as weapons. So jabs refer to one male using his gonopodium to jab the other male, and they'll jab the other male in various parts of the other male's anatomy. So these are not erroneous mating attempts. These are what appear to be, they're jabs all over the body, so they appear to be aggressive interactions and happen in the context of aggression. And then blocks, which involve one male following a female and potentially displaying to her and keeping her from another male trying to interfere with that pair. So that's what blocks are. We subjected these data to a factor analysis. And I want to highlight a couple of the factors because these are the ones that ended up being significant. I'm not going to talk about all of the analysis we did here, but I'm happy to talk with you about that afterwards if you're interested. So one of the most important factor was mating activity, and then aggression to the other male was also important. So I want to talk about those. So first I'll show you that factor 1, mating activity. So these are the trials with those females who like plain morph males. Right, in the association time test. In those scenarios, the black morph males had much higher mating activity than the plain morph males. However, with the females who didn't seem to care too much, the mating activity was very similar between the two morphs. So perhaps these data suggest that the black morph males, with their persistent displaying to females and following females really closely all the time while they're trying to display to them, that persistence may overcome female preference for plain morphs in that group. Right? That persistent following and displaying is something that plain morph males just almost never do. And not surprisingly, again, given what I've said about the pleiotropic melanocortin receptors, black morph males are also more aggressive than plain morph males. This is aggression directed at the other male. And so this suggests that there may be some advantages to black morph males having to do with male-male competition and not sexual selection in the form of female choice. Right? And that turns out to be quite important in this system. We did also undertake some investigation, and this is still in the early stages of another graduate student who just has started, to see if there's any evidence of negative frequency dependent mate selection happening, sexual selection happening in the system. So This is the work of Sam West and her team, some of whom are shown here. So far, 165 undergraduate students have worked in my lab in the last 12 years. So I'm only showing you some of them, right, just to give you an indication. So what Sam hypothesized is that if females were habituated to one morph, so housed solely with males of one morph for some period of time, in this case several weeks, those females should prefer the opposite morph in mate choice tests if there is a rare male effect. So females are selected to bolster offspring diversity by mating with rare male morphs. That can maintain polymorphism and explain polymorphism maintenance. And if that's going on, then females habituated to one morph should prefer the opposite morph. So she has started undertaking this study. Again, this is still somewhat in progress. We're still increasing our sample size, but she had one treatment group where there are females housed only with black morph males, visually separated from all other tanks. Those fish were allowed to interact with each other directly, so the males were allowed to mate with the females, etc. And then other tanks where there were only plain morph males in with the females for a period of several weeks. And then these fish, and I should mention here, these are all from the recently acquired Cuban stock. We are moving towards working only with those fish for probably obvious reasons, being concerned about inbreeding and things like that. So she, after the several weeks of treatment phase, she then subjected these females to mate choice tests. So again, the standard dichotomous choice test. However, she tested the females with unfamiliar males. So neither of the males that a female saw were males that she was housed with during the treatment phase. Those were unfamiliar to her. And the two males were also not ever housed together. So they were unfamiliar to each other as well. Performed these association time tests, and here's what she and her team found. Contrary to the idea of negative frequency dependent selection, females preferred the morph that they were housed with. So females in the black morph treatment preferred black morph males. Females in the plain morph treatment preferred to associate with plain morph males. So these data point to, if anything, positive frequency dependent selection. This could not maintain the polymorphism as such, right? And it's not surprising to me that females may have learned to prefer the morph of male they were housed with. Because of the mating strategies that these males employ and how different they are, females would do well to anticipate the kind of mating behavior that males are going to direct towards them, and as such may prefer the males that they're familiar with. I want to point out these females had the opportunity to mate with males of both morphs in their history before they were used in this experiment. So they weren't like the females who had never seen plain morph males before, or the females who had never seen black morph males before, right? So this is only that 3-week habituation, and I think what's going on here is that females are mitigating the costs of harassment by males. So a lot of studies have been done on Poeciliid fishes and harassment by males and the negative effects on female fitness of males persistently trying to sneak copulations or court the females or follow them around. It minimizes the chances of females being able to forage successfully. It actually negatively impacts their fitness. And for example, this paper by Dada was included Gyrodinus metallicus and showed that females school together more tightly when males are present, suggesting that they're trying to protect themselves from this mating behavior. And remember that, again, the black morph males persistently follow females almost all the time and constantly try to mate with them, even as the females are trying to forage or whatever else. And in contrast to that, the plain morph males lurk, chase, and sneak to copulate with females. And this was described way back in 1980 by Farr, who thought there was only one morph and looked at plain morph males and described their behavior. So other authors have described their behavior other than us. And I want to emphasize that some of the costs of harassment for females include not just missed foraging opportunities, but also injury that can occur. So this is what the tip of the gonopodium of Gyrodinus metallicus males looks like, right? Poeciliid males, it's not just a tube. These genitalia have co-evolved in a sexually antagonistic co-evolution. They've co-evolved to circumvent female choice. And so some of these spines and things may be used in male-male competition, because we know that this species uses their gonopodium as a weapon in fighting. But structures like these palps are thought to be holdfast, to try to hang on to the female for longer than she is selected to let males hang on and try to transfer sperm that way. And that this could be an adaptation in that context. I'm being vague in my wording because in this species, people haven't shown that yet. We don't know that. It's a hypothesis for why this evolved. Females are not without their countermeasures, right? When you're talking about the arms race between the sexes, this is Apologies for the giant genitalia pictures, but this is the female genitalia, right? When I ran this talk by my lab, they all just sort of went, whoa, when I put these up. But so here's the anus of the female. This is the gonopore, the genital opening of the female. These structures are thought to have evolved to prevent males from being able to inseminate females more easily. So this may be what the males are holding on to as they're trying to copulate with the female. Even just a microsecond longer could be what it takes for males to be able to transfer sperm to females. But this may pose an impediment to forced copulation on the part of males. So everything I've shown you so far seems to put the black morph males at a disadvantage, right? Females either have no morph preference, or they prefer the plain morph males unless they were very recently habituated to black morph males. In the wild, the black morph males are very rare, so you're not going to have a scenario in which females are habituated to black morph males. What Rodet Rodriguez and his colleagues have seen in Cuba is that there's no site that they have surveyed that has an abundance of black morph males. They're rare everywhere. So that means that that scenario wouldn't happen in the wild. So under the typical scenario in the wild, females are probably going to have either no morph preference or a preference for plain morph males. So why do males do this display? And as I said earlier, I and my colleagues and other people have called this a courtship display, but now I'm not so sure that it's a courtship display, at least not in the context of sexual selection in the typical way that we think about this. Here are some older papers in which people described the display of these black morph males. All of these authors called this courtship display in their description of what happened. But we think now that maybe this is a species indicator display, which would make sense because these black morph males are so rare. Females would rarely encounter them. They may not know that they're G. metallicus. And so following the females around really closely and continually trying to mate with them may be the only way that they can have mating success, given their rarity. So that's one possibility. We haven't tested this idea, but I think I would like to in the future. The other possibility is that this is part of an aggressive mate guarding repertoire on the part of these males. Right? Black morphs are more aggressive than plain morph males, and it's possible they're indicating to other males, I've either just mated with this female, or I'm going to mate with this female, I'm going to keep trying to mate with this female, so stay away. So that's the another possibility. So we wanted to know, is there sexual selection operating in the system via female choice or male-male competition on a more nuanced scale? So within the black morph males, is there sexual selection acting on their colored traits? And this is the work of Erin Willian and Crystal Castillo, a former and current grad student of mine. And I want to emphasize that these males are not black and white. They're black and yellow, right? So keep that in mind as I talk about the work that we've done. So one of these studies involved measuring morphological traits, the area of ventral black on both sides of the body, gonopodium size, body size, a whole bunch of different traits. So we measured those traits, and then we performed the dichotomous choice test, similar to what I've been talking about already. In this case, there was a slight modification. We had a dichotomous choice tank set up that was with porous barriers. So the fish could not only see each other, but they could smell each other as well. So chemical cues were potentially being exchanged in this test. Once again, males were paired to minimize body size differences, and in this case, we used those ventral black area data to maximize the difference between the two males in the pair for how much ventral black they had, to see if females preferred males that had more black. After the dichotomous choice test, once again, we performed direct interaction tests involving all three of those fish put together in a tank to look at what happens when they could interact directly and to introduce male-male competition into this system as well as female choice. The upshot of what we found is that aggression is key to mating success in this species. Farr found something very similar for the plain morph males as well. And so what these data are showing is that when we took the males within each pair and scored them as being either the dominant male or the subordinate male based on how much aggression was directed to the other male and what proportion of that aggression was directed by that focal male. What we see is that the dominant male in each pair gets to display more to the females, so follow the females around and display more, and makes more copulation attempts. So they have more access to mating opportunities the more aggressive they are. And let me show you what aggression looks like in this species, because I haven't yet. So here are a couple of males. They're biting each other. They tail slap each other. Notice that instead of safely holding the gonopodium up against the body, right, instead they extend that gonopodium and show it off to each other. So I think there's some interesting work to be done here, more fine-scale work on gonopodium size and characteristics in the future to see what exactly are they signaling to each other by holding those gonopodia out. All fights are not quite this intense. Sometimes it's just a male chasing another male away, and that's it. That's the entirety of the aggressive interaction. So we found that blacker males are more aggressive. And this is one data set. We have multiple data sets now that confirm this result in different groups of males. The more ventral black area the male is covering the male's body, the more aggressive he tends to be. So then we asked, well, is there a relationship between which male the female chose in the association time tests and how much mating activity that male exhibited later on in the direct interaction tests? In other words, could females tell which male was going to be the winner later on or determine which male was going to be the winner later on? We can't distinguish between those two. We found that there was, in fact, a positive relationship. So males that the females spent more time next to in the dichotomous choice tests also got to display more, follow her around and display more, and also got to make more copulation attempts. So I'll just emphasize here again, we don't know if females are somehow cueing in on which male is going to be the more aggressive one later on, or if they're making that male more aggressive by choosing him in this dichotomous choice test, because the males could see each other and smell each other during the dichotomous choice tests as well. So within the black morph, blacker males are more aggressive and seem to succeed more in terms of mating activity. So then Erin, for her master's thesis, wanted to look at not just the ventral black area, but also the yellowish body regions of these fish. Depending on the lighting during photography, you can really see this yellow more in some cases than others. But when you look at these fish, up close in a tank, you can see that they have quite a golden yellow color. Some authors have described them as being lemon yellow in color. So it depends on lighting conditions. But we, in this study, looked at body size, gonopodium size, all the measures I've already talked about, ventral black area. We also looked at dorsal black area because they do have some dorsal spotting, so we scored that separately. And then Erin and her team also measured the hue, saturation, and brightness of a set of homologous points shown here on each of the males in this study. And for those who may not know, hue refers to, in general, what we would call the color. So is it red, blue, green, something like that? That's what hue is, measured on a color wheel, as degrees on a color wheel. And then saturation refers to really how intense the color is. So it's sometimes also called chroma. And it's a measure of how much gray is mixed in with that color. And brightness is a measure of how much either white or black is mixed in with the color. So if it is low enough, if there's enough black mixed in, then all colors converge on black. And depending on the hue, colors will tend to look more white the higher the brightness is. So both brightness and saturation are measured as percent values, and then hue is measured as the degree on the color wheel. So once again, I wanted to, we subjected these morphological data to a factor analysis as well. I want to emphasize that we measured the hue, saturation, and brightness from digital images that we took of males that were unanesthetized and held in small, really tiny plexiglass containers. Because Erin has shown that these anesthetics, and other people have shown this as well, anesthetics cause these fish to darken, and we didn't want that to confound or otherwise impede our measurements of hue, saturation, and brightness. So here are the results from the factor analysis of these morphological variables. And we decided, although all of these are interesting and important, we decided to use yellow saturation as a measure to pair males. So yellow saturation should be an indicator if this yellow coloration is pigment-based, and we don't know that. Erin tried really hard to do some measurements of the skin of these fish to see if she could figure out whether it was carotenoid pigment-based or not, and we just couldn't do it with the time that she had and the constraints that she was under. So we don't know for sure, but we thought yellow saturation would be an important aspect of the yellow coloration. So we decided to pair males, minimizing body size difference and maximizing the yellow saturation difference between the two males in each pair for this study. And so Erin did that, she and her team, and they also employed a slightly different dichotomous choice test than the others I've told you about. She was concerned about the males being able to see each other and smell each other and potentially influence each other's behavior during the dichotomous choice tests. So for her test, she constructed a tank design that looked like this. So the male and the female could see each other in each case, but the males couldn't see the other male, and they couldn't smell, no one could smell each other. All of these barriers were clear glass with no perforations. And so based on this design, we found there was no effect of coloration on female choice. Females don't care how black the males are, they don't care how yellow the males are, they just don't seem to care about anything to do with male traits or which morph they are. There was also no relationship to subsequent aggression or mating activity of the males. So it's possible that the males need to smell each other or see each other in the dichotomous choice test in order to later on for that to be associated with how they behave in direct interaction. She did do direct interaction tests again, and what she found again bolsters this notion that aggression is what's key to mating success in this species. The yellower of the two males was more aggressive in her pairs, significantly more so. I don't have a slide for this, but she also found a positive relationship between the yellow brightness and aggression as well. So the more yellow these males are and the more black they are, the more aggressive they are, and the more they seem to succeed as a consequence. So no evidence for female choice, but mounting evidence for male-male competition being important. So given that, we wanted to know whether females maybe directly prefer aggressive males. Maybe it's not the blacker males or the yellower males or anything else, but rather maybe females prefer aggression directly. So I want to emphasize in this video that when males are aggressively interacting with each other, they will do so happily when females are watching, right? So females have the opportunity, she's getting a chance to eat too, which is nice. Females have the opportunity to look at these fights between males and potentially assess who the winner is and maybe choose that male as a mate. So we examined this phenomenon, which is called social eavesdropping. So if there is social eavesdropping operating in the system, then females should prefer to associate with the more aggressive of two males, but only after watching them fight, right? So they should be gaining information by watching males fight. So this study was performed entirely by undergraduate students, and there was no graduate student overseeing this. And so it was exciting that they did such a great job on this study, and here's what they did. So now we're looking at dichotomous choice tank from above in this case. We went back to this sort of standard design for dichotomous choice tanks because we were a little bit concerned that we didn't know for sure if the female could see both males simultaneously. I don't want to diss Poeciliid females too much, but it's possible that she saw one male, went around the corner, saw the other male, and forgot about the first one, right? We're a little bit concerned about that. So I don't know that we've hit upon the perfect design by any means, but this is a very standard type of dichotomous choice design, so we decided to go back to that for this experiment. So the students conducted a female choice trial. Again, the males size matched, tested randomly which male was on which side was randomized, and they always performed 2 trials, switching the males in between to ensure that we minimized side bias. And then they performed what we call an eavesdropping trial. They put the 2 males together and let them fight it out with the female watching. So she could see them and they could see her, but they couldn't directly interact with the female. Under these conditions, the males were super aggressive, way more aggressive than they are when they can actually mate with the female, which makes sense, right? So they don't have the opportunity. They tried really hard. We were a little bit concerned, actually. They kept bashing against the glass trying to get to the female on the other side. But eventually they figured it out and they just duked it out, between them. So very aggressive. And we performed another mate choice trial afterwards to see if the females preferred maybe the winner of this aggressive interaction. And in fact, we did find some evidence that there was a positive relationship between how aggressive the male was in that eavesdropping trial and the association time for females, but only in that second trial. So in the first trial, we found no such relationship. It was a flat line. However, after the females watched the males fight, they spent more time near the winner of that fight. So all of this taken together is interesting. It points to maybe some role for intrasexual selection rather, in maybe maintaining this polymorphism, right? Maybe because black morph males are more aggressive, they're maintained in the population, but maybe only until their frequency gets higher. Maybe when their frequency reaches a certain point, they end up not doing as well, and that keeps black morphs at a low frequency. That's a possibility. And so moving forward, I would really like to focus a little bit more on what's called indirect genetic effects. This is an area of behavioral ecology that emphasizes the genotypes of the social group that an individual finds itself in. And so under indirect genetic effects, the genetic variation of social partners influences the phenotype of the focal animal. And it's very possible that regardless of which morph male we're talking about, the frequency of the morphs around that male may influence his fitness, his behavior, his success. Furthermore, there may be a G by G interaction such that this effect is different for plain morph males and black morph males, right? And so going back to that eastern mosquito fish work, there's been some really nice work done even more recently showing just these kinds of effects in that species. And so that is part of what spurred me to start thinking about things this way. So one of the things that I did very recently is to compare across data sets to see what kinds of behavioral differences emerge when there are two black morph males together with a female versus a plain and a black morph male together with a female. Is there a difference in these different types of social groupings? And what I found is, and I looked at this across a range of behaviors, but what I found was the only significant behavior was copulation attempts. And these are copulation attempts during which the male visibly makes contact with the female. These are the copulation attempts that are most likely to actually be successful and transfer sperm, right? And what I found is that when it was a plain and a black morph male together, there were a lot more copulation attempts than when it was two black morph males together. There was no significant morph effect. In other words, all males are enjoying potentially greater mating success when there is a mixed morph pair than when there are two black morphs together. And I think this is largely driven by the fact that when there are two black morph males together, they're spending a large proportion of their time aggressively interfering with each other, following the female around, and fighting with each other, and not able to get as many actual mating attempts in. So this is, again, very preliminary work, and I'm looking forward to making more formal comparisons of this, right? I did this by combining data sets as opposed to setting out to do this study, but I'm looking forward to doing that in the future. There is another entirely undergraduate student performed study that I want to talk about that may have some relevance in this context. So these students, Alex Wilcox and Evelyn Teodoro, wanted to see whether they could uncover potential patterns of negative frequency dependence such that the black morphs frequency and fitness increase when they're rare and then decline when they start to get more common, and there's this sort of oscillating pattern maintaining them in the population but at low frequencies. And they tested it, in this case, by looking to groups of stimulus fish. So far, all the dichotomous choice tests I've told you about is a female with one male on either side. This may not be the most realistic scenario, right? So Evelyn and Alex decided to test females with groups of males on either side in that dichotomous choice test. And they predicted that females should prefer mixed morph groups over a monomorphic control group of only plain morph males, with the idea that mixed morph groups offer females the opportunity to increase offspring diversity because there are different types of males within that group. However, based on our previous data, they predicted that females should still prefer plain morph biased groups because they seem to exert either no preference or a preference for plain morph males. So here's what the dichotomous choice tests looked like in this case. So on one side was a control group of 4 plain morph males, and then on the other side was a treatment group of varying frequencies of the two morphs. So they tested the control against a group of all black morph males, 3 black to 1 plain, and 1 black to 3 plain. These 3 ratios. They subjected the females to this. Again, they randomized which side the control was on and performed 2 trials per female so that they minimized side bias. And this is also a data set that involved females raised with both male morphs so that they had a life experience with mating with both male morphs, or at least seeing and interacting with both male morphs. This is a data set we're building on still, but so far, here's what we found. So When the treatment group was all black, there was no difference, essentially, between the amount of time females spent with the control versus the plain black group. And when there was a mixed morph group that was black biased, so 3 black morph males to 1 plain morph male, there was no significant difference in how much time the females spent with them. So basically, these data are pointing to no morph choice on the part of these females, which is what we found in the past as well. However, when there was a mixed morph group that was plain morph biased, right, so mostly plain morph males but 1 black morph male, females did tend to prefer to associate with that group. So these data hint at the idea that maybe there is some negative frequency dependence going on in the system that's maintaining these black morphs in the population, but rarely, as a rare occurrence because of, possibly because of their aggressive interference, possibly because of their persistent following of females and harassing them. But these may both come into play. Now, one important aspect I haven't talked about at all that is a glaring omission is predation, right? It is very likely that predation has some role in the maintenance of this type of polymorphism. This is one of the main predators in Cuba that occurs in G. metallica sites. This is Nandopsis tetracanthus, the Cuban cichlid. And this is an image we took of a fish we collected in Zapata Swamp. And this video is, let's see if I can get this to play. This video is another predator that co-occurs with these fish. This is a introduced predator. It's the blue tilapia, Oreochromis aureus. Here is a female guarding her fry. So here are the fry, and there's the female. I show you this video to show you what this other potential predator looks like, but also to give you an indication of what the bottoms of some of these sites look like, right? How kind of heterogeneous and junky they are. It's unclear to me in this kind of a setting which of the 2 morphs may be more visible to predators. In the lab, the black morph males stand out incredibly more clearly. We can easily see them from a distance, and plain morph males are very hard to spot. But in this kind of an environment, I'm not so sure what the prediction would necessarily be. But this requires field tests of predation to really see what role predation has, if any, in this system. So to summarize what I've talked about, these black morphs persistently display and follow females around. They're more aggressive, whereas plain morph males lurk, chase, and sneak copulations with females. Females either prefer plain morph males or exhibit no morph preference, and both of those would put the black morphs at a disadvantage in the wild. So the display that the black morphs almost exclusively perform may function in species recognition or mate guarding, or both, but it doesn't appear to represent courtship display so far as we are able to uncover at all. Yellower and blacker black morph males are more aggressive, which seems to be the key to mating success. And females appear to eavesdrop on aggression and choose the more aggressive male. The higher male mating success in mixed morph pairs and the female preference for these plain, biased, but mixed morph groups suggests that there may be a role of negative frequency dependent selection in the system, potentially contributing to the maintenance of the polymorphism. It sounds like we're at the end, but there is a fly in the ointment here. I've shown you these 2 morphs. It turns out there may be a 3rd morph, and it may look like this, all right? So this is the yellow morph G. metallicus. We've known about the yellow morph for a number of years because we've had them in our lab, but we got them from hobbyists. And the yellow morph males, here's another image, set of images. These are yellow morph males in our lab. We obtained them from hobbyists. We thought, because Rodette Rodriguez and our other Cuban collaborators had never encountered these, we thought that maybe they are entirely an artificially selected phenotype that occurs outside of Cuba only, except that very recently, Rodette and his advisor Ingo Schlupp found a yellow morph male in the wild. We are also finding some rumors online that this is what we have to rely on for species like this, that in a certain part of Cuba, the very northern part in Veradero, there may be populations where yellow morph males occur quite frequently. And that's the really disturbed area, lots of golf courses and resort hotels and things like that. So it is possible that there's something going on there, maybe released from predation pressure, that contributes to a higher frequency of yellow morphs, and that Rodette and his colleagues just hadn't sampled those sites. We don't know. So 1 male has been found in the wild, but clearly it was a yellow morph male. The other thing we know about this morph is that when you bring plain morphs into captivity, as Rodet did in Cuba, he brought a bunch of plain morph fish, males and some females, into captivity in Cuba in his lab that he had set up in his house in Cuba. And after a couple of generations, he started to see males appear that look like this. So again, they're being fed maybe higher carotenoid food because they're in captivity. They are released from predation. We don't know exactly what the environmental change is, but he saw this in more than one male. He saw several males appear that look like this. So it's intriguing. It's possible that maybe even in the wild, there is something more complicated going on with this. Maybe it is a trimorphic scenario. Maybe there's a rock, paper, scissors game happening, at least in some sites. It's certainly not going to be widespread because they would have seen yellow morphs with all the extensive sampling that they've done. They would have seen yellow morphs more frequently if they were distributed throughout Cuba. But certainly in some locations that haven't yet been surveyed, this may be what's going on. And this kind of thing is not at all unheard of in Poeciliids. This is my favorite Poeciliid currently. This is a pentamorphic Poeciliid, Poecilia pare. These are all male morphs of this species. It has 5 male morphs. And this polymorphism is maintained by a complicated set of selection pressures. Female choice favors the carotenoid-based coloration of these morphs. The blue morph tends to be more visible in certain microhabitats and enjoys a mating advantage in that context. These barred black morph is really aggressive, and that's how they get reproductive success. And then there's a female mimicking sneaker morph that sneaks copulations, right? So very complicated, involving spatio-temporal selection pressures varying, different microhabitats, negative frequency dependent selection, female choice, all kinds of things, all contributing, predation also contributing to this. So the G. metallica scenario may be much more complicated even than we already know that it is. And so in the future, there are a number of things that I would look forward to doing with collaborators. So this is Rodet again, and this is his current PhD advisor, Ingo Schlupp. And we want to describe the behavior and look at all 3 morphs together and see what kinds of other correlates of behavior exist. Things like relative testis size and sperm production. Is there a difference? Are plain morph males devoting more of their energy towards sperm production for sperm competition than the black morph males are, for instance? And then we also plan, in collaboration with Dr. Jean Davidson in our department, to do some genomics and characterize the MC1R sequences, but probably more fruitful will be gene expression studies with that locus to see if at least the black and plain morphs differ. Maybe all 3 morphs we'll take a look at. And then, of course, we hope to get to Cuba to do field work on this system and look at more frequencies, look at predation and things like that. That's contingent on a lot of things, practical constraints. Some of the things that, I said how rewarding it is to have a job such as the one I have, but there are some practical constraints. So taking undergraduate students to Cuba is one of the constraints, right? So it's difficult to do, but we're hoping to get there and obtain maybe predation risk assessments for each of these morphs. So I would like to acknowledge these collaborators. I'd like to bring things back to all of the wonderful students that I work with, which again are the best thing about my job by far, and these funding agencies for their support. And thank you so much for coming today.
Time for a couple of questions.
Thanks for the great talk.
Of course, thank you.
I'll ask some later, but
sure.
This one is, so do the aggressive males do other things? So you showed us that they, everything in the context of mate choice, but I guess you showed that there's parental care from the mother. I don't know if the fathers also participate then.
There's no parental care at all.
Right, and then so could the more aggressive ones be better at finding food or better at other things or worse at other things, and then is it heritable?
Right. And we don't know the answers to those questions, but they're incredibly aggressive over food. So this species, unlike others that I've worked with, like guppies, for instance, this species is incredibly aggressive. Females are aggressive to each other, males are aggressive to female, everybody is aggressive to each other. So we can't do any behavioral observations within about an hour to 2 hours after we feed them because they're just, trying to kill each other, basically. So everyone. Females, males, everyone. And so that's intriguing and interesting. So yes, it would be really interesting to look at we've done some studies of aggression over a defensible food source, an algae wafer. We have those data, we haven't looked at them yet, but yeah.
As you were talking about, I've been involved with working with Poeciliid fishes off and on for a very long time.
Okay, cool.
And And what I have always wondered is the biomechanical question. The gonopods don't go in very far.
Right.
The males are presumably ejecting a packet of sperm.
They are. It's a spermazoigmata.
From the
Male to the female.
Into the female.
Yeah, that's a really good question.
It's a hydrodynamic issue.
Yeah, absolutely. So from what I understand, it varies among species whether this occurs or not. So in some species, the males deposit that packet of sperm outside the female. And the sperm, I don't know, make their way somehow into her. Sometimes they may attach it to the outside of the female, and there are some, and Greg, you may have heard this, there's some reports of males, of sperm release happening entirely not on the female, and sperm making their way into the female. And then in other cases, the males do manage to insert that sperm packet into the female's gonopore in that very brief time. There's also variation in terms of how long the males hang on during copulation among species, right? And probably among individuals as well. Yeah, it's a good point. And I hadn't heard until recently about this, the idea that they sometimes deposit that packet on, but not in the gonopore, or maybe even not even on the female at all.
So when you were looking at the sort of social environment effects and so on, you noticed that the number of copulations went up for males. But did that actually produce more babies? And the broader question is, do females produce more or less, depending on who mates with them, whether they have choice in mating,
right,
and so on and so forth? So how much are the females controlling how many
offspring they have? So the short answer, just like with the heritability of aggression, the short answer is we don't know yet, right? We're just starting. Not much has been done. I mean, we're not just starting to work on them, but not much has been done on this species. So I would be shocked if there wasn't post-copulatory sexual selection going on. Shocked if there wasn't. So absolutely, our best gauge for how successful a male is copulation attempts with contact. But that's not at all the same thing as offspring production, fertilization, any of those things. So one of the constraints we're under is that undergraduate students don't stay in the lab very long, right? They finish, they graduate. Without a PhD program, it's really tough to do breeding experiments, which is what is sorely needed to see put females with males of both morphs and see who has more offspring would be a great thing to do. So that's a longer term kind of project that I would have to be the one to do, because there's not any one student that could pull that off. But good questions.
Can I have another question related to this one? Are attempts from black males, so basically aggressive attempts, are they less successful than sneak attempts?
Again, in other species, we know that is the case, that sneak copulations have a lower success. So in species like guppies, for instance, sneak copulations have a lower success than copulations preceded by display. But we don't know. For these fish, we don't know if the black morph's copulation attempt, if you compare apples and apples, do the black morph males, are their copulation attempts more successful or not?
My point was, if it's not to display, but just trying to pin down the female is actually defensive.
Is she defensive against the sneak copulation attempts? Is that what you're saying? Or? She doesn't have as much opportunity to be defensive against sneak copulation attempts because
aggression
from males. The black males don't tend to be aggressive to the females in the context of mating. They're aggressive in the context of foraging. So if we're feeding them, they're really aggressive to each other in general. The black morph males don't tend to be aggressive towards the females. They are persistent. I mean, they harass the females, but they're not aggressive. It's the plain morph males that tend to zoom out of nowhere and chase them around and copulate with them.
Hi, Gita. Very nice talk. I was just curious about the black morph males. Now, how much is the natural variation in blackness? You said that the females prefer the ones that are darker, but how much real variation is there that females can distinguish?
Good question again. In the wild, how much real variation is there? Because we have these lab populations that we're looking at, and they're quite variable, but we don't know in the wild how much variation there is per se. However, look at their display, right? They're swimming around, showing off that chin, showing off that black. It is variable for sure, as with all traits. So I think the females can see how much black the male has. It's also about how much is filled in the center and how much it extends to the gonopodium. So I think females should be able to pick up on the variation that exists even in the wild from having looked at those males. But
is there a great deal of variation? Do you know how much roughly what that variation is?
Not off the top of my head, yeah, but there is a great deal of variation. I'm prepared to say there's a great deal of variation, especially in terms of how much of the gonopodium is black. Yep. Any questions?
Hi.
Hi.
Quick question. I was really
Sorry.
Sorry. Oh,
Sorry, I didn't mean it.
interested in the fact that you said that the blacker males or yellower males were more aggressive. And I was wondering if you looked at anything like chromatic boundary strength or like transition intensity, because I could imagine being very black and very yellow at the same time would have a greater chromatic
Contrast.
and perhaps make an individual more conspicuous.
We haven't looked at that. It's a really cool idea. I actually jot those down because I haven't heard of it described that way. Maybe the black is an amplifier of the yellow, for instance, right? I think that's really intriguing, and we already have the data in hand potentially to look at that. So maybe I can chat with you afterwards about how, it sounds like maybe you know about how to compute that, because I had not heard of that. But because we perform factor analyses, we integrated all of these, but that may not be the best way to look for that, what you're talking about, to do with that contrast. But that's a good idea. And that may be why they're black entirely. The black may have evolved to set off the yellow. Right? Yep. Pretty cool.
Cool. Let's thank Gita again. And
Thank you.